Hi there, and very welcome to this short explanation of track number 1 for the Mitosis Domain Generalization MICCAI Challenge, or short MIDOG 2025. Now, my name is Marc. I am one of the organizers of the challenge, and in this short tutorial I will briefly go through what the particularities of track number 1 of the challenge are and what you should know about it. As I said, there is a track number 1, so there is also another track, in this case our track number 2, and the participants can choose if they want to participate in either one of them, both of them. Yeah, of course we're really happy if you participate in both of them. Now let's get right into track number 1. In track number 1, as in track number 2, it's all about mitosis, and in this case it's about the detection of mitosis. So mitosis is the process of cell division. It contains several stages which look quite a bit different in the H&E stained histopathology slide, and actually I did a video about that already, which considers our track number 2. So in order to watch that, if you're interested, please have a look there. I'll link it just up there. Oh, well, maybe there. Okay, regardless, I'm not good at this yet. What is MIDOG track number 1 about this year? It is about the detection of mitosis across domains. So already in the past we had several different kinds of domains, and it was always about the generalization. Here you can see some examples, some histopathology examples of cutaneous mast cell tumor, of breast cancer, of lymphoma, different kinds of tumors, and it was always about the detection of mitosis, because mitosis is a tumor marker that is very interesting for all of those cases. What is new in this iteration of the 2025 MIDOG challenge? Now, I'll just briefly go through the history, where first MIDOG challenge was about the generalization to unseen scanners. Scanners alter the way an image looks significantly, and thus the first challenge was solely concentrated in the field of human breast cancer, and it was basically to have known scanners and then generalize to unknown scanners. The second version of MIDOG, MIDOG 2022, was about the generalization to unseen tumor types. So we had a bunch of tumor types and we wanted to generalize to 10 more tumor types. So that was what the challenge was about. Now, we do have some more tumor types also in MIDOG 2025, but the major novelty in MIDOG 2025 is the generalization to whole slide images. Now, what that means, I will tell you in brief next. Before I do that, I just want to highlight that due to resource constraints, we will not apply the algorithms on whole slides, but we will apply them on representative sections of the whole slides. But why do we do that? In order to showcase this, I have here some examples. So this is an example of the TCGA breast cancer cases, and you can see here in green, it's maybe a little bit hidden, you can see it here and here and here, and you can see those detections of mitotic figures. Now this works quite well. It is a detector that was trained on the so-called MIDOG++ dataset, which I will detail a little bit later, it is basically a case of, well, it was trained on regions of interest, on dense tumor areas, and it is now applied on regions of interest, dense tumor areas again, so that works quite well. However, if we just go to the same slide and just go to another area, maybe here or maybe even, just a little bit over there, this is a necrotic area. And in this necrotic area, there are no mitosis, but we can see a lot of false detections of mitosis, because there are some patterns that are pretty similar of mitosis. So as you can see, our detector that we trained on this data set, well, has quite some false detections, and that's undesirable, especially in the application to whole slide images. So we've seen this for cases like inflammation, for necrosis, for other kinds of morphological patterns, and thus we thought, that's the final frontier for mitosis detection. It's generalization not only to different tumor types, to different scanners, it's also towards the whole slide, towards a real clinical applicability. So that's what this MIDOG challenge is about, or at least the track number 1 of this MIDOG challenge is about. Now, how can you imagine the challenge to be set up? One thing that was always a little bit, I wouldn't call it unique because other challenges do it as well, but that sets us apart from some challenges is that in MIDOG you never have access to the test data, so to the test images. Not even the input images. So that means that we will centrally process all the algorithms. We will use our good partner grandchallenge.org for this again, and the participants need to submit Dockerized algorithms for that. So we do this in order to avoid kind of overfitting on test image data. Because knowing of the test image data could help you to, like adapt the algorithms to actually what should be unknown data. So in order to not have any, in order to have a level playing field for everyone, no one has access to that. It will be the algorithms that have access to that during inference. What these algorithms have to do is basically they will get a region of interest spanning 2 square micrometers, around about 7,000 times 5,000 pixels, and they will have to detect mitosis within these and provide them in a JSON compatible format. We do provide a reference container that also gives a reference algorithm as an implementation, so the participants can see how it is expected, or how the algorithms are expected to yield the results. As you can see already here, it is about retrieving some points. Also here already visible is that the points are not in pixels, but they are actually in millimeters. Are they in millimeters? Yes, I think they are in millimeters. And in image coordinates, and then we want for each detection a probability in order to be able to also calculate secondary metrics. Yeah, that's how this whole task is set up. This whole thing works. What datasets can you use? Well, first of all, we have the MIDOG++ dataset, which is, well, at least in terms of MIDOG challenges, a new dataset. It was already released in 2023 by our group, and it contains basically all MIDOG datasets so far and adds some more data to it. Well, it's still pre-selected regions of interest that were assessed to be like diagnostically meaningful in the tumor. It contains like totally 12,000 roundabout mitotic figures. It's 3 blinded raters consensus, and it's 503 tumor cases. So quite a lot in terms of data diversity. Then we also have the MIDOG WSI-CMC dataset, which is a canine mammary carcinoma, so breast cancer dataset. It contains around about 14,000 mitotic figures. Same thing, 3 blinded raters. And in this case, it's only 21 tumor cases. Why? Because it was whole slide images that were annotated, fully annotated. And of course, that takes quite a bit more time, so you cannot easily annotate like 500 cases or so. And then finally, we have another tumor type, which is also a canine tumor, which is the Mitosis WSI-CCMCT dataset, which we also made available already a little bit back, and it contains around about 45,000 mitotic figures, again 3 blinded raters, 32 tumor cases, and it's also completely annotated whole slides that are available in this dataset. Why would you want to use those? Well, I mean, I think clearly the novelty in MIDOG 2025 is generalization to whole slides, so it cannot hurt to have some whole slides completely annotated. Right, what's next? As also already described in the track number 2, so soon we will provide also for MIDOG 2025 the Docker container with the reference algorithm that we provide here in this context, and then from August 15th onwards we will allow the submission on the preliminary evaluation set. So in order to be able to do that, please register on grandchallenge.org and get your account validated. Secondly, or lastly, we will have on August 30th and 31st, we will have submissions of on the actual test set. You can only do that if your container already worked successfully on the preliminary evaluation set, so make sure that you calculate time for that a little earlier. All right, so that is what I wanted to say for now and what I wanted to share for now. If you're now interested in that, you want to have maybe a look at our webpage, midog2025.deepmicroscopy.org. If you have further questions, please don't hesitate to get in touch with us on our Discord server. The link is on the webpage, and I think also somewhere here down there in the comments. I guess. All right, so have a good time. See you at the challenge. Bye bye.